Welcome to the PubMed Daily Review for March 10th. Today we scanned 3,504 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, a druggable redox switch on SHP1 controls macrophage inflammation by Maying Ng and Edward T. Chuchani. Let's break down its key contributions. Many diseases involve inflammation driven by immune cells like macrophages. While proteins are key players, most are hard to drug. We're learning that tiny chemical changes, called redox modifications, on specific protein parts called cysteines, can powerfully control how these immune cells behave. This research asks if we can find these redox-sensitive spots and use them to develop new medicines. To find these druggable sites, the researchers used advanced techniques called deep redox proteomics. This allowed them to map out hundreds of cysteine sites that change chemically in immune cells. They then focused on a specific protein, SHP1, and found a critical cysteine, CYS102, that acts like a switch. They designed a special molecule, SCA, that can precisely attach to this cysteine, turning SHP1 on and reducing inflammation. The team discovered a remarkable 788 in vivo redox-regulated cysteines across many immune proteins. Crucially, their new molecule, SCA, selectively bound to SHP1's CYS102. This targeted action effectively reduced key inflammatory signals, like IRAK signaling, and suppressed the production of pro-inflammatory cytokines in response to LPS in both mouse and human macrophages. This demonstrates a powerful new way to control inflammation. This research is exciting because it reveals a completely new way to potentially treat inflammatory diseases by targeting specific cysteine modifications on immune proteins. They've not only found a druggable target on SHP1 but also created a roadmap of other potential targets. This work paves the way for developing highly selective drugs that could offer new hope for patients with conditions driven by chronic inflammation. Up next, our second paper out of 5, higher proximity of PD-L1 plus tumor cells to PD-1 plus tissue-resident memory CD8 determines response to neoadjuvant chemoimmunotherapy in non-small cell lung cancer by Emin Zhao and Xiaorong Sun from Department of Nuclear Medicine, Shandong Cancer Hospital and Institute, Shandong First Medical University and Shandong Academy of Medical Sciences, Jinan, China. Let's break down its key contributions. Neoadjuvant chemoimmunotherapy is a game-changer for resectable non-small cell lung cancer. However, we still struggle to accurately identify which patients will benefit most. While PD-L1 expression is a common marker, it's not always reliable. This study explores whether the physical closeness of immune cells to tumor cells can provide a more precise way to predict treatment success. Researchers looked back at 44 NSCLC patients who received neoadjuvant chemoimmunotherapy. Using a technique called multiplex immunofluorescence on pre-treatment tumor samples, they meticulously mapped the distances between PD-L1 positive tumor cells and various types of T cells. They then compared these spatial relationships in patients who responded well to treatment versus those who didn't. The results were striking. Patients who responded to treatment had significantly closer distances between their PD-L1 positive tumor cells and several T cell types. Most importantly, the proximity of PD-1 expressing CD8 plus tissue-resident memory T cells to PD-L1 plus tumor cells emerged as the best predictor, with an AUC of 0.743. This spatial relationship was strongly associated with a better treatment response, showing a significant odds ratio. This study highlights a crucial insight: it's not just about having the right immune cells or PD-L1 expression, but where they are located relative to the tumor. The physical closeness of specific T cells, particularly PD-1 plus CD8 plus TRM, to PD-L1 plus tumor cells appears to be a powerful new predictor for neoadjuvant chemoimmunotherapy response in NSCLC. This finding could lead to more refined patient selection strategies in the future. Up next, our third paper out of 5, PD-1 genetic fate mapping uncovers immune cell diversity mediating the efficacy of combined PD-1 blockade and chemotherapy by Ayuko Yamaguchi and Takashi Ebihara from A Department of Medical Biology, Akita University Graduate School of Medicine, Akita, Japan. Let's break down its key contributions. We know that combining chemotherapy with immune checkpoint inhibitors like PD-1 blockers can be very effective against certain cancers. However, PD-1 can be expressed on many different immune cells, and its expression can change during treatment. This makes it hard to pinpoint exactly which cells are driving the anti-tumor response. This study aimed to use a clever genetic trick to track these PD-1 expressing cells and see what happens when we combine these two powerful treatments. To tackle this question, the researchers developed a special mouse model where they could genetically tag cells that express PD-1. They then treated these mice, which had lung tumors, with either anti-PD-1 therapy alone, chemotherapy alone, or the combination. They used advanced techniques like single-cell sequencing to analyze the immune cells in detail, tracking their identity and function throughout the treatment. The results were quite striking. While PD-1 blockade alone had limited success, the combination therapy with chemotherapy led to a marked improvement in controlling the tumors. They identified 15 distinct types of immune cells, and importantly, saw an expansion of cytotoxic CD8 T cells that were capable of killing tumor cells. This suggests the combination therapy is really revving up the immune system's attack. So, what does this mean for patients? This study highlights that combining chemotherapy with PD-1 blockade can be a powerful strategy, working by broadening the immune system's attack. The genetic tools used here are also incredibly valuable for future research, helping us find the hero immune cells that are key to treatment success. This opens doors for developing even more targeted and effective cancer therapies down the line. Up next, our fourth paper out of 5, a novel eye-tracking digital marker outperforms plasma biomarkers in detecting cognitive impairment by Yi Ling and Benyan Luo from Department of Neurology, First Affiliated Hospital, School of Medicine, Zhejiang University, Hangzhou, China. Let's break down its key contributions. Detecting cognitive decline in older adults is a major challenge. Early identification is key to managing conditions like Alzheimer's. While plasma biomarkers show promise, they have limitations. This research investigates a new, non-invasive digital tool, eye tracking, to see if it can accurately identify cognitive impairment. This study enrolled 119 cognitively normal individuals and 157 with cognitive impairment. Participants underwent digital eye tracking tests. Their performance was compared against established plasma biomarkers and cognitive assessments. The main goal was to evaluate how well the eye tracking tool could distinguish between cognitively impaired and unimpaired individuals. The results were striking. The eye tracking panel achieved an area under the curve of 0.865 for classifying cognitive impairment. This significantly outperformed plasma biomarkers, with p-tau217 showing an AUC of 0.769 and A beta 42/40 at 0.699. Other biomarkers like GFAP and NFL also performed less effectively than the eye tracking tool. This indicates eye tracking is a highly promising method. This study highlights the significant potential of eye tracking as a novel digital biomarker for cognitive impairment. Its superior performance over plasma biomarkers suggests it could become a valuable, non-invasive tool in clinical settings. Future research should focus on validating these findings in larger, diverse populations and exploring its integration into routine screening protocols. Up next, our fifth paper out of 5, Signal Peptide Engineering and Codon Optimization to Enhance Alpha-Gal A Activity for RAIV Gene Therapy of Fabry Disease by Siwoo He and Yuqing Chen from National Clinical Research Center for Geriatrics and State Key Laboratory of Biotherapy, West China Hospital, Sichuan University, Chengdu, China. Let's break down its key contributions. Fabry disease is a serious genetic disorder where the body can't break down certain fats, leading to damage in organs like the heart and kidneys. While enzyme replacement therapy exists, it has drawbacks. Gene therapy, particularly using RAAV vectors, offers a potential solution, but we need to make the therapeutic enzyme work even better. This study explores ways to boost the effectiveness of gene therapy for Fabry disease. To tackle this, the researchers first tested different signal peptides in liver cells, HepG2, to see which one best helped the therapeutic protein get secreted. They also codon-optimized the gene itself to improve its expression. Promising candidates were then packaged into RAAV8 vectors and tested in mice that model Fabry disease. Various assays were used to measure enzyme activity, protein levels, and the reduction of disease-causing substrates. The results were very encouraging. They found that one engineered signal peptide, SP21, increased enzyme secretion by over twofold in cells. Codon optimization alone boosted activity by fourfold. But the real power came when they combined both strategies. In Fabry mice, this combined approach resulted in a nearly seven-fold increase in enzyme activity and, crucially, effectively cleared the toxic buildup of the disease substrate, lyso-Gb3. This study demonstrates a powerful dual strategy to significantly improve RAAV gene therapy for Fabry disease. By optimizing both protein secretion and gene expression, they achieved remarkable results in a mouse model. This combined approach holds great promise for developing a more effective treatment. Future research will focus on translating these findings into clinical applications.